In this video, we're going to discuss the genetic code and the effects of mutations. We're going to begin with a very brief review of the transfer of genetic information or protein synthesis. Remember, proteins are synthesized continuously within a cell as proteins are needed for growth, reproduction, metabolic processes, and more. Protein synthesis occurs in two steps. In the first step, we take a molecule of DNA and we copy it through a process known as transcription into a molecule of messenger RNA. Messenger RNA then carries that code from the DNA to the ribosomes, in which then the process of translation occurs. In translation, we use transfer RNA molecules as well. Transfer RNA molecules carry with them each an amino acid. Transfer RNA will then use the genetic code found on messenger RNA to know how to align those amino acids into a particular sequence of amino acids, which will then make up the protein. So let's review for just a moment when we refer to the genetic code. Remember, the genetic code is based off of nucleotide bases, and for RNA, those nucleotide bases are A, U, C, G: adenine, uracil, guanine, and cytosine. Okay, in DNA it uses thymine or T. RNA it uses the U or uracil. So in order to come up with a genetic code, we take three of those nucleotide bases or triplets and we form them into a codon. So look here for just a minute. You'll see on messenger RNA we refer to three nucleotide bases as a codon. On transfer RNA, three nucleotide bases are referred to as an anticodon because they're going to be the mirrored image or opposite of the codon. On DNA, the three nucleotide bases are referred to as a triplet. So there's many different ways to know that genetic code. We're going to base our reading of the genetic code off of the messenger RNA and the codon. And so we use codon charts, one such as this. And if you orient yourself to this chart, you'll see that we have the three nucleotide bases. The first one you find from the left-hand side, the second one by scrolling over the top, and the third one you come down to the right-hand side. So let's go back just a moment and look at our three codes and kind of review that chart. So the first codon was AUG. If we look here at our chart, we find A, scroll over to U and find G, and we see that codes for methionine. Methionine is also referred to as the start codon because it begins all of our protein synthesis. Let's go back and look at the second. The second codon is UUG. If we scroll down to our chart again, we find UUG, and it codes for leucine. The third one is CAA, and it's going to code for C. Scroll over and we find AA, glutamine. And the cell will continue doing that process in order to be able to come up with that unique amino acid sequence that forms that protein. So another thing we need to point out about this chart, though, is that there are three codons that are referred to as stop codons. Stop codons are the UAA, UAG, and UGA. And when you have those codons, it tells the cell where the polypeptide chain needs to stop and end that protein synthesis process. Okay, so now it's also important for us to discuss what happens when this process is not accurate. So when we go through the DNA replication process, if that replication is not accurate and you cause a change in the genetic code, then that is going to change what happens when the DNA information is passed to the messenger RNA. And then that leads to possibly an incomplete or an inappropriate amino acid sequence. So we need to transfer the genetic code accurately. So a mutation, if a mutation occurs, that could be detrimental. So let's talk a little bit about mutations. Mutation is a permanent change in the nucleotide sequence, and that's going to occur when the DNA replicates itself. Okay, so during the DNA replication process. There are two different types of ways that mutation can occur. One is a spontaneous mutation, and that occurs when it's during the normal replication process. The other one is in induced mutations, which then that causes, because of a mutagen or even a carcinogen, which is a chemical substance, or UV lights, can then induce a change in the nucleotide bases. So Mutations, again, they're permanent and they will be passed along to the daughter cells and can be detrimental to the cell. So let's look at different types of mutations. In our first type of mutation, only a single base pair is affected, and those are referred to as point mutations. And there are three types of point mutations. The first is a silent mutation. In a silent mutation, the mutation results in the message coding for an amino acid, which is the same amino acid as found in the original code. Therefore, there's no change, and the protein sequence doesn't change. So no impact on the cell. In the second, it's a missense mutation. In the missense mutation, the mutation results in a messenger RNA coding for a different amino acid. That's going to then change the polypeptide sequence and overall possibly the protein. The third type of point mutation is a nonsense mutation. In a nonsense mutation, the mutation results in the messenger RNA coding for a stop codon prematurely. That will then cause the protein to be shortened or truncated, and usually the protein then is becoming non-functional and leads to a serious negative impact on the cell. So let's start by looking at these three mutations. Okay, so this is our original strand of DNA, and notice here during replication I have indicated in red where the mutation has occurred. All right, so in this case the DNA went from AAC to AAA. What then results is a change in the messenger RNA during that transcription process, and we went from what was supposed to be UUG to UUU. And if you go back to that codon chart, you'll notice that UUG was supposed to code for leucine, and due to the mutation, we have UUU, which now codes for phenylalanine. So in this case, it is a missense mutation because the mutation results in the messenger RNA coding for a different amino acid sequence. And depending on that amino acid will depend on the impact it has on the protein. So let's look at the second one. In here, we have our original DNA again. Again, you'll see here where we have a point of mutation. And in this case, we went and changed a C for T. So the sequence went from AAC to AAT. So messenger RNA was supposed to code UUG, but now codes UUA. The end result is that UUG coded for leucine, and the new mutation UUA also codes for leucine. So this is what we refer to as a silent mutation because the mutation results in the messenger RNA coding for the same amino acid. So this probably is not going to impact the cell at all. The third type of mutation. In this type of mutation, again, here you're going to see we have the original strand of DNA, the point of replication of where the mutation occurred. And here we went from AAC to ATC. And if you look, the messenger RNA was supposed to code for UUG, but now codes for UAG. The original protein should have coded for leucine, and now it codes for stop due to the mutation. So this is a nonsense mutation, and because of the fact that the mutation results in the messenger RNA coding for a stop codon prematurely. So this protein is going to be truncated and probably going to be non-functional, and this one will lead to a detrimental effect usually on the cell. Now there's another type of mutation known as a frame shift mutation. And in a frame shift mutation, either one or two, a couple of base pairs will be deleted or inserted. That then results in a change in the number of bases, and therefore when the messenger RNA gets made, it too will have a different number of bases, and then that means the reading frame becomes altered. So let's look at our example here. So here's our original DNA, and if you'll remember, it codes for messenger RNA such as this, which is the leucine, glutamine, serine, valine. So in the case of a frame shift, notice what happens during the replication, and here I've coded it in red. So what we did there is we inserted a thymine. So we inserted a base, and then that then leads to a shift right here. So if you look at the resulting messenger RNA, right here where I've got it outlined in red, we start to shift the way we're reading that sequence, and what that then does is results in a different amino acid sequence. So we have start leucine on both of them because the point mutation occurs here at the glutamine. So in our new messenger RNA, glutamine now becomes threonine, and notice from that point, because every frame shifts at that point, every amino acid is also going to be different, and this will then lead to an entirely different protein, and again can be very detrimental to the cell. So with that concludes this, because we've gone through a review of the protein synthesis process and the genetic code, and then looked at different kinds of mutations, the 3 types of point mutation and the frame shift mutation. Look at D2L for more information and let me know if you have questions.